very short break between.
Yeah. Should we wait or should we get started?
Finished quite late. Yes.
Recording in progress.
Okay. Can we start, Olga?
Yes, sure. Let's start.
Okay. All right. So good morning, good afternoon, and good evening to everyone. Welcome to this last session of the day. My name is Gaby Lee. I'm an associate professor in the Department of Physics at Florida International University. So I'm presiding this session, and I will be giving the first talk. So I will share this screen. Oh, okay, hold on. Okay, so first I'd like to thank the organizers for the opportunity to present. And so I'm going to be talking about our recent work about using optic two-dimensional coherent spectroscopy to study interatomic interaction and coordination in atomic examples, including cold atoms. So as we all know that usually ultra-fast femtosecond lasers are not used to study ultra-cold atoms since they are intrinsically incompatible in their characteristic timescales because ultra-cold means ultra-slow. So the question we ask ourselves would be, what can ultra-fast nonlinear spectroscopy actually do in atomic examples, especially in cold atoms? And also, is it even possible to actually perform ultrafast nonlinear spectroscopy using femtosecond lasers in a cold atom cloud? And I'm hoping to answer at least these two questions in this talk. So in this talk, I will basically discussing our work of using 2D spectroscopy to study dipole-dipole interaction and many-body correlation in atomic examples. First, we did this in atomic vapor in vapor cells, and also then we did this in a cold atom cloud. And hopefully in the future, we will be able to do this in an atomic array, which will be more deterministic. So this topic of studying dipole-dipole interaction in atomic examples, especially in atomic vapors, and had a really long history. And in the past, people actually used, for example, the different techniques. For example, in the frequency domain, selective reflection, and in the time domain, using the time-resolved methods by using quantum beating spectroscopy or transient four-way mixing spectroscopy. Now, all of these techniques before, they actually rely on either measuring a large enough energy shift or broadening due to strong dipole-dipole interaction in the sample, which actually requires a really high density atomic vapor. To put that into perspective, so here I actually plot the atomic number density as a function of the temperature in a vapor cell for both potassium and rubidium. So of the previously recorded techniques, they require at least 10 to 15 per cubic centimeter number density, which corresponds to a vapor cell temperature higher than 200 C. Therefore, those techniques are actually, it will be very difficult for those techniques to actually detect any effects of dipole-dipole interaction at lower temperatures. And just to compare, so a typical number density of a cold atom cloud prepared in a typical MOT will be at the order of 10 to the 10 per cubic centimeter. So all of these techniques will be really difficult to actually do anything with the cold atoms. Therefore, what we're going to use is a technique called optic multidimensional coherent spectroscopy. Now, the basic idea of this technique actually came from AMR spectroscopy, and in 1993, Shao Mukmil wrote a paper, and he actually proposed that we can actually do similar experiments in the optic domain by using femtosecond lasers. And it took a while for the technique to actually develop. But by now, this technique has been pretty well developed with all different types of approaches, and it has been used in different systems to look at, especially many-body effects and quantum dynamics in those systems. And my postdoc advisor, Steve Candiff, and some other former members in the group, we actually recently wrote a book on the topic, which was published this year. So that will be a pretty quick summary of the technique and its applications, especially in the systems that physicists are interested in. Okay, so the particular 2D spectroscopy technique we are using in this experiment is something called double quantum 2D coherent spectroscopy. A simple explanation can be explained by using a 3-level matter system, a ground state 0, single excited state 1, and then double excited state 2. So what we do is we actually send a sequence of pulses to excite the system. So the first 2 pulses will create a quantum coherence between the ground state and the double excited state 2. And this double quantum coherence is going to oscillate at the double frequency of the excitation laser. Therefore, the name double quantum 2D spectroscopy. And then the third pulse can actually convert to that double quantum coherence back into a single quantum coherence, which can be detected or either emit a signal which will be at the single quantum frequency. So by using this technique, what we are able to do is to actually detect the double quantum coherence during the second time delay if we scan the second time delay by detecting a single quantum signal. So we're actually detecting a higher quantum coherence by detecting just a single quantum signal. So in the experiment, what we do is we scan the second time delay, capital T, and measure the signal in the third time delay and report it that in the time domain, and then we can perform a Fourier transform. Then we will get a double quantum 2D spectrum in the frequency domain. Okay, so the signal of that spectrum actually relies on the existence of the double excited state, which is state 2 right here. Okay, so now we apply this to, for example, a potassium atomic vapor. Now, what happens in the potassium atomic vapor is that if we tune the laser to excite both the D1 and D2 lines of the potassium atoms, then the double frequency of this laser does not really hit any atomic transitions. The closest energy states are 4D and 5P, which are actually quite far away from the laser spectrum. So we don't really have a double quantum state in this case. However, if we do the experiment, we will see pretty nice double quantum signal coming out from the experiment. A hand-waving explanation for this is that in order to get the signal, we have to consider the two-atom picture. That is, if we consider 2 atoms together, for example, if I use two level atoms as an example, then in the 2-atom picture, we'll have a ground state, which both atoms are in the ground state, and we'll have a single excited state where one atom is excited and the other atom is in the ground state, and we also have a double excited state. So then in this case, we now have a double excited state. However, that won't be enough to actually give you a signal because the system will be totally symmetric and all of the pathways will actually cancel out, resulting in no signal. In order to get a signal, we also need some type of interaction between the 2 atom states that will introduce an energy shift for the single excited state and double excited state, which the interaction will also introduce a change in the dephasing rate from the double excited state to the ground state. That actually breaks the symmetry and then the contributions from all of the pathways will not cancel out entirely. That will give you a signal. So in the two atom picture, so what we're looking at right here is in this spectrum. So the x-axis is a single frequency, single quantum frequency. The y-axis is a double quantum frequency, which is corresponding to the double quantum coherence during the second time delay. Now for potassium, since we have the three different combinations of D1 and D2 transitions for the two atom states, so now we actually have two different energies in the double quantum frequency. So what we're seeing right here are corresponding to these correlated two atom states, or you could say these are two atom decay states that are showing up in the double quantum spectrum. But remember, we also need interaction between the two atoms. Therefore, this technique is a sensitive detection for both the interatomic interaction and the two atom correlation. Now, just really quick. So we just really quick. I just like to mention that we could also do this in rubidium. Now, the difference in rubidium is that we have a two atom state as well as a single atom state, which is within the laser spectrum. So we actually see three different resonances in the spectrum, and this one right here corresponds to the two atom states, and these two, they're actually from single atom states, which is sometimes is useful because you can actually use a single atom states as a reference to study the dependence, because we understand how to calculate the response from single atoms. We could also do this not just from for S and P states. We could also absorb the two atom states of these states. Just really quickly to show you the results. So these are the spectrum. This is the spectrum from the these states. Okay, so that was for two atoms. Now you could ask, what about three atoms? Can you actually see three atom states? Now if you consider three atom states with both D1 and D2 combinations, okay, we're now back to the potassium. So for the D1 and D2 combinations, you will have four different combinations for the three atom states. And in order to excite now not the double quantum coherence, but triple quantum coherence from the ground state to the three atom states, now we need an even higher order excitation and a higher order linear spectroscopy experiment. So to do that, theoretically, it's very simple. You just need more pulses to do a higher order experiment, right? Then you will be able to absorb the triple quantum coherence. Experimentally, so in some of the geometries, it's actually pretty hard to get more pulses. The geometry, experimental geometry we use is a collinear geometry. So all of the pulses actually propagating in the end, propagating in the same beam, and we actually distinguish the pulses by tagging each pulse with an LM frequency. Now, the trick we use here is that we actually reuse some of the pulses, because each pulse can always be considered two pulses with a zero time delay. So now if we reuse some of the pulses, then we can easily do a higher order experiment. We just need to detect the signal at a different reference frequency, which will be corresponding to a higher order signal. Okay, so that's a experimental technique. So we went ahead and did try this for three atom states, and for sure we saw the signal from three atom states, which has four resonant energy levels in the triple quantum axis. And then a week later, we tried even higher order experiment, and we tried to look for the four atom states, and we saw that. And we tried even higher orders, 5 atom states, 6 atom states, and then we went all the way to 8 atom states. So what we're seeing here is that this is a higher order nonlinear experiment where we actually see the correlated multi-atom states, and we can choose which number of atoms we would like to see. So that could be very handy if you now would like to study the dependence of some many-body property as a function of the number of atoms. Okay, now all of these experiments I have shown you, they were actually done in an atomic flavor. Okay, so they were not done in the cold atom. Now, there's one problem with that is, well, you actually have some ambiguity in the experiment because you're not sure exactly how many atoms you are and what is the distance between the atoms. You have a statistic explanation for that, but it's not deterministic. So an ideal experiment would be if you can just grab two atoms, or for that matter, you grab any number of atoms and you can actually put them in an array, and then you use this technique to study that. Okay, so that can be done with an atom array. But before we even try that, the first question we ask ourselves is that, well, that would be a pretty hard experiment. Can you actually do that? So why don't we just try this in a cold atom cloud first? Can we even get a signal from the cold atom cloud? That would be a good start, right? So so there are immediately two questions if you do this in the cold atom. So the first question is the frequency resolution, because in this technique, we're using a femtosecond laser. You don't really have enough frequency resolution to resolve, for example, all of the hyperfine structures. So, for example, the data I showed you, they don't really show you the hyperfine structures, right? So now that problem was actually solved by Steve Candiff group. So they showed that you can actually use a frequency comb to do 2D spectroscopy, which will give you enough resolution to resolve all of the hyperfine levels. Now, another question is the detection sensitivity. Do you have enough sensitivity to actually do this experiment? Well, here's what we tried is, well, it turns out 2D spectroscopy is really sensitive. So I come back to that chart I showed you before. So where we could do is basically we could actually get a signal at the room temperature with a vapor cell. So if you think about that in terms of the interatomic distance, so that density actually corresponds to a distance of about 60 microns, which is quite surprising. So with that, we were pretty confident that we have enough sensitivity to do this. So we set up a cold atom setup with basically the standard MOT, and then we combine the 2D spectroscopy setup with the MOT together. And we started by looking at some one-dimensional spectrum, for example, pump, probe, and full mixing, and we were able to get pretty nice signal. And then we went ahead and tried 2D spectroscopy. We can try different types of 2D scans, the so-called 1 quantum, 0 quantum, and double quantum scans, and we were able to get a pretty nice signal with very good signal-to-noise ratio. So we're quite confident that now we can actually perform 2D spectroscopy in cold atoms, and now we're continuing on doing some more measurements in the system. Now, in order to actually be able to do this in an atom array, another question will be the number of atoms, right? So can we actually do this for a single atom? So what we did is we just focus the beam really tight, and then we reduce the atomic density, and then at some point we should just on average get one atom in the beam. So we did this dependent study. So this curve corresponds to a single atom signal. This red corresponds to the double two atom signal, and we can show that the two atom signal actually disappears a little bit earlier than single atom signal. Then that's where the transition going from two atoms to one atom. So we are now kind of know that this probably will work for single atom as well. So if that works, that will be our plan in the future. So I know I'm running short of time. We'll skip the summary. I just would like to thank the people who did the work in the group. And the last word is just advertising that I'm looking for a postdoc in our group. If you are interested in, or someone that's interested in, please email me. And thank you, and I'm open for questions. So, okay, so I'm sorry, I'm trying to see if there's anything in the chat.
Hi, guys.
Yes, Dimitri.
Yes. Hello. Sorry for the small delay.
We can hear you. Go ahead.
Yeah, I'm very sorry. Just give me a few minutes and I'll be ready to launch. I apologize.
No problem. Let me know whenever you are ready, okay?
Yes.
Mm.
Okay.
Okay, let me do the introduction first.
Yeah, okay, sounds good.
Okay, great. So the next speaker is Dmitry Krosky from Texas A&M, and he will be talking about tip-enhanced Raman imaging of plasmon-driven coupling of 4-nitrobenzene serial on gold-decorated magnesium nanostructures. And he's getting ready with his slides.
Yeah, so sorry. If possible, I want to change the topic of my presentation and make it slightly more biological. So I know that this topic, the subject interests a lot. The can you see the screen?
Yeah, can you put it in the presentation mode?
Yeah, okay. I know that this topic interests a lot the Institute on Biophotonics. So it's something very interesting. That's something my lab works on the past 5 years. Specifically, we develop laser-based sensing for confirmatory diagnostics of biotic and abiotic stresses in plants. And the question to ask is, what is overall this sensing? And the answer is relatively simple. What we try to do, we try to develop the sensors that can be used directly in a field to bring the information from the field. And the information about what happens in a field can be used to improve farming approaches, can be used to make plants essentially bigger and better, and ultimately mitigate the stresses that plants experience, because these stresses that are divided into two groups, they're called biotic and abiotic stresses. They can reduce the crop yield by 30% if we talk about biotic stresses, and 70% if we talk about abiotic stresses. And we also work in the area of precision nutrition and personalized diet with this sensing technology, primarily targeting the food we eat in the food we buy in grocery stores and in a supermarket, and food we consume in restaurants. So what we want to do is to develop the sensor that could read macronutrient composition in that food, and essentially will help us to balance the nutrition and avoid diseases that are linked with to nutrition. And also, ultimately, we want to develop the sensor that can be used to personalize the diet, because it's known that different individuals that have completely different response to the same food. For one person, eating, say, a burger may be nothing, but for another, it can be quite filling. So what we ultimately want to do is to personalize the diet. But let me first walk you through our findings and tell you how it actually works and what we found. So the problem with agriculture is essentially the growing world population. More and more people need food every day, and currently we have around 1 million people starving, and this number obviously will only increase. And one strategy to fix this is to increase agricultural land, but obviously it's not very exciting, because sooner or later we'll turn the whole planet in a farm field. The alternative way is to do it better, and doing it better, we can have an improvement in machinery, which is already advanced quite a lot. I'm sure you agree there is a big leap between John Deere's plow and what is known as John Deere now. The food chemistry that is used as the fertilizers and pesticides, etc., but also in plant genetics. There are lots of new breeds that are used at this moment, and these breeds are more resistant to pathogens and more resistant to various abiotic environments. But what the actual problem is, we don't have information coming from the field. So it's kind of a one-way approach. And what we want to do is to make it two-way approach, is for us doing something to the field and for the field to communicate it back to us. And of course, this is possible if we have sensors working in the field. And like I said, the sensors can be quite useful. And some of the sensors already exist. For example, soil sensors that can be linked to the iPhone, and the farmer might not even need to show up in the field if the temperature is not appropriate, and show up precisely when the temperature will be good. But of course, we want to look at the plant health and develop sensors that can be used to detect and identify biotic and abiotic stresses. And like I said, biotic stresses are quite bad. They drop the crop production about 30%, but abiotic stresses are far more detrimental. And if we can sense the stresses, that we can apply fungicides, water, and fertilizers that will cease the spread of pathogens and essentially provide economic advantages to the farming. Now, when it comes to sensors, talking to many farmers in different states, including Texas, I found that what farmers want, they want the sensors to be inexpensive, fast, portable, and pretty much accurate. And if we look at the currently used methods in plant sensing or overall diagnostics of plant diseases, we can find that there are quite a few, and they can be divided in two groups: molecular methods and imaging methods. Molecular methods is PCR and ELISA, and imaging is RGB imaging, thermography, and hyperspectral imaging. Now, if we look at how they fit to these four categories, we find that both PCR and ELISA, they're very expensive. Yes, they're fast and portable, to some extent accurate. PCR is more accurate than ELISA, but the problem is the cost. The cost of PCR is about $25 a sample. ELISA is $15. It's way too expensive for farmers. If we look at the RGB imaging and hyperspectral imaging and thermography, we find that the direct cost of these methods is very low, because they're not consume any chemicals. However, they're not fast, especially when it comes to hyperspectral imaging, and they're also not accurate, because what they sense is the change in the plant color, which obviously doesn't mean anything, because the plant can get yellow from 5 different reasons: from drought or from fungal infection. And this is the problem. So as you can see here, essentially there is not a single method that can be used to satisfy the farmers. And the method we propose is called Raman spectroscopy. It's named after C.V. Raman, an Indian scientist. And I'm sure I don't need to talk about this method too much for this audience. I just want to remind that it's based on inelastic light scattering. We shine a laser light on the sample. We collect electromagnetic radiation that is scattered off, and we cut off the elastically scattered photons with optical filters. We collect inelastically scattered photons, and dispersing their energies, we get a spectrum, and vibrational bands in the spectrum correspond to the chemicals that are present in the sample. By other means, using Raman spectroscopy, we can probe the chemical composition of the sample of interest, and it's exactly what Alma and Art already described. Now, the beauty of Raman spectroscopy is it can be miniaturized to the handheld devices, and as you can see, there are two, but we possess two, but there are many quite nice handheld instruments that can be brought directly in the field, and also it can be used as a standoff spectral acquisition. Here, we use a telescope. We shoot the beam that comes from the laser, and the scattered radiation is collected by the telescope. Now, let's look how Raman fits in this category. First, there is no direct cost associated with this, so direct cost is zero. Also, it takes only 1-2 seconds to get the spectrum, so it's really fast. And we can put the chemometrics that required to predict the spectra directly in a software and get the result right away on a screen that you can see, for example, here on this instrument. Now it's also portable because they are handhelds. And finally, I want to spend the next several minutes to convince you about or to demonstrate the accuracy of Raman spectroscopy, how it can be used to detect and identify bacterial and other diseases. The story I want to tell you is actually about Texas, the plants that are grown here. And these plants are citrus plants, and they're grown in 3 counties, primarily down in the south. And the problem is that they can be infected by bacteria called Candidatus libibacter that once get in the plant, clots the phloem, and the phloem stop transporting nutrients. So plant essentially start dying. The problem is by PCR, it can be diagnosed. But if you look at the number of trees, we talk about hundreds and thousands of trees. So, of course, farmers are reluctant to use these methods because they cannot afford to spend so much money. Now, we did an experiment in which we tested the accuracy of Raman spectroscopy. We looked at the symptomatic trees that have some yellowness because of lack of nutrition, the pre-symptomatic plants that were tested positive by qPCR, however, have not yet any symptoms, and also plants that have yellowness in leaves. However, this yellowness has nothing to do with the pathogen themselves, and this yellowness is coming from nutritional deficiency, and of course, a control group of plants. And what we found is the following. If we look at the healthy trees, first we get an outstanding spectrum in just a second of acquisition, and we can clearly see all vibrational bands that correspond to macronutrients. I will talk about this in a second. But if we look at the both pre-symptomatic and symptomatic trees, you can see big changes in several vibrational bands. But the most exciting is the increase in about 1601 and 1630 bands. We know that they come from aromatic. And also around 1440 and 1455, these two come from CH2 vibrations. Finally, if we look at the trees that essentially nutritional deficiency, we have a completely different spectrum. Again, it's quite different intensity of the band around 1600, and we have a new band at 1247. Now, when we do chemometrics, because looking at the spectra is not enough, so in my group, we collect dozens, if not hundreds of spectra from every class, we can predict the accuracy of identification of healthy versus nutritional versus HLB with, as you can see, on average 98% accuracy. And if we're interested to pull apart healthy versus early stage versus late stage, we can do it with 100% accuracy. Now, the question is, what does actually Raman sees? To answer this question, to interpret the vibrational bands, which is very necessary for plant biologists, we do HPLC analysis of these plant leaves. And through HPLC analysis, we were able to identify molecules that change in the plants. And then we were able to acquire the Raman spectra to demonstrate that we actually see a change in that specific molecular analytes. Habin, how much time I have?
You have 10 more minutes.
Okay, very well, very well. So
including questions. Okay. Yeah.
Okay, I'll be quick. So what you can see is, again, changes in the vibrational band that come from 1600 and 1525. So we know that the 1525 band comes from carotenoids and 1600 come from the aromatics. So I'll skip HPLC and I will tell you that aromatics is a very important set of molecules in a plant that are responsible for plant defense. And it is known that the change, as a result of the plant pathology, the concentration of pericomeric acid increases a lot. And when we get the spectrum of pericomeric acid, we observe vibrational bands exactly in this region, which demonstrates that we see change in the concentration of pericomeric acid in the plant. But also, we observe changes, according to HPLC, in viatexin and dismethin. And when we acquire the spectrum, again, we see quite a good match between the standards and the Raman spectra acquired from the plants. Now, we also did this work on carotenoids, and what was very exciting is carotenoids have very specific frequency of vibration around 1525. So we know that our vibrational bands are centered at 1525. However, looking at the standards, we can exclude several carotenoids that are also present in the plant. And what means that highly likely we see the change in the concentration of lutein and violaxanthin in the plant. And to prove it, we did HPLC, and indeed, we see a strong decrease in the concentration of lutein in the infected plants. So hope I was able to demonstrate you the set of both biochemical and spectroscopic studies that are interconnected, because Using Raman, we can detect and identify different stresses through through changes in the vibrational bands. But what is very important for plant biologists is to understand what we detect, and this kind of symbiotic interaction is very helpful to establish this and develop this method for confirmatory diagnostics of biotic and abiotic stresses in plants. And again, I apologize for the small delay. I'll be glad to take questions. Thank you.
Okay, great. We have plenty of time for questions. Maybe I will get started. So the data that you're showing there, they were taken in the lab. Okay? So what does it take to actually for you to go from the lab to the field?
Well, so all the spectra were acquired with the handheld instrument. So we actually did this work directly in a field. The case study I showed you, it was done in a field. We had to travel to Veslaco, it's South Texas, and collect spectra directly there. The only thing where we cheated, we took the leaves from the trees and acquired spectra in a lab only because it was very hot. It was done in July. But we also did work directly in the field here in College Station. We work exactly with plants that grow in the field. And don't really mimic any type of stressors. So we're already there. We work in a plant, in plants that are grown in a field, and either simply because of the environmental conditions, prefer to do measurements in a lab rather than stay in the field.
Okay, great. So in the interest of time, so we'll move on to the next talk. So the next talk will be given by Hans Schuster. I don't know if he is here.
I don't see him here, so since I'm also a co-author of this work, I will give a talk. Yeah.
Okay. So then we will have Alexander Klemensky to give the next talk. Actually, next two talks. The first one will be hyperfine structure of xenon by frequency cone calibrated saturation and collinear fast beam spectroscopies. Please go ahead.
Okay, let me get to this.
And this talk is 15 minutes. So we actually, we're not seeing the correct screen of yours.
Can you see my screen?
Yeah, but it's a presenter's mode. We're not seeing the presentation mode.
Let's see.
It's better.
What do we see? What do we see?
Well, this works too, if you don't mind.
This works. Okay.
Yeah.
So I will go on then.
Yeah.
Yeah. So this work is about measurements of hyperfine structure of xenon, assisted by frequency comb calibration, and it was done at Texas A&M. And motivation for this work is that a hyperfine structure reflects interaction of magnetic field of electrons with nuclear dipole moment and magnetic moment and quadrupole electric moment. And we, in this talk, will consider measurements of hyperfine structure of xenon. Then we discuss this method of Doppler-free saturation spectroscopy, and this will be done with TISA-Pi laser, tunable in the range from 820 to 841 nanometer. The application of frequency comb for these measurements will be discussed, and we will present results on this hyperfine structure and also isotope shifts that were measured with this technique. And finally, I will also briefly discuss a different method which uses fast ion beam laser spectroscopy and also allows measurements with Doppler-free results. So here are the transitions that were studied, and they start with excited state. So to get there, we need something like 7-8 electron volts from the ground state. And to enable this, we have to produce RF discharge in xenon. Now, the problem in these measurements, they require like precision spectroscopy, is that transitions are Doppler broadened because, all atoms are moving chaotically. Now, we use approach which is quite well known. It's related to lamp tip measurements and was pioneered by Lamp, and also some work was done by Hench later, where we have two laser beam, one pump, one probe, counter-propagating. So for the probe beam, if the Doppler shift in the opposite direction, so to employ the saturation effect, we need actually Doppler broadening for both beams be the same, and this is happening just at the some class of atoms that move perpendicular to the beam, and then we see some reduction of the absorption by the probe beam. Here is the experimental setup. So we have a pump beam coming to the our cell with the discharge, and we have also probe beam that propagates in the opposite direction, and we use different wave meters to measure approximately the frequency of the transitions. And of course, the signal to noise ratio can be improved significantly by using lock-in detection. Now, to enable like calibration with the frequency comb, we use beating of our TIE laser with rainbow comb that is produced at the repetition rate about 80 megahertz, and pulses are very short, about 8 femtoseconds. So the spectrum of this comb is quite broad. So then we select some beating, and as the laser is scanning, we use the following trick. We mix the bit node with the mixer, shifting it by like quarter of the repetition rate. And as the laser frequency is scanning, we are getting spikes in the signal, each f repetition rate divided by 2, which gives a kind of ruler in the frequency domain and allows to exactly determine the frequency. And the frequency comb is reference to a rubidium standard for stability. Now, here are some results for 823 nanometer transition. So we have 4 transitions for xenon 129, and we have 10 transitions for xenon 131. Here on the right-hand side, we see the measured spectrum, and below we see the schematic showing the calculated positions of the peaks for xenon 129 with black color and xenon 131 with red color. Below is the absorption of the direct pump beam, which shows that due to Doppler broadening, we don't have this resolution that we need. And interestingly, in the middle, we have this strong dip which is absent as a signal here in the upper one, because we actually supersaturate the sample, and this effect will be discussed a little later. Here is an example of another transition, 840, and here we have 3 transitions for xenon 129 and 8 transitions for xenon 131. And the number of transitions, of course, determined by the allowed transitions and like interaction of the angular momentum of the atom of electrons and momentum of the nucleus. And for the odd isotope of xenon, we see this splitting and get hyperfine structure. For like interpretation of the observed signals, we used this saturation model, for, which also described in the book by Sigman. And this formula for the profile of the absorption peak, it takes into account also saturation intensity, and depending on this relation, the profiles can be quite different. So here are some simulated shapes of these absorption signals at different parameters, and we varied like line widths, the portions that send to pump, and the probe beams, and optical density. All of them, to a certain extent, affect the shape, and therefore what we actually observed in our experiments can be qualitatively confirmed and explained with our model. Now, here are results for measuring of hyperfine structure constants. So when we measure these positions of the hyperfine lines, we can make some system of equations that will relate to hyperfine structure constants. And from this, we can determine these constants and compare to other to results of other groups. And I will show this a little later graphically. Now, for even isotopes, we see isotope shifts, which correspond to like usually detuning close to 0. And these isotope shifts also put in the table and compared to results by other groups, and we have good agreement. And now another approach to measuring hyperfine structure can be based on the technique that is called fast ion laser beam spectroscopy. And acceleration of xenon ions reduces Doppler shift, because we have this effect of velocity bunching in this case. And then as we scan the laser wavelengths, we can tune to different transitions of the hyperfine structure, and then using fluorescent signal, we obtain this spectra. So this the setup itself is shown on this slide, and it has many functions. We need to accelerate ions, then make overlap of the ion beam with the laser beam, and then adjust the velocity of this ion in the post-acceleration region it's fine tuning to make this interaction close to the detection spot, and then we use this fluorescence detection. So the schematic of this transition is shown here of this experiment. So we start with metastable level, and it's produced actually in the ion source. So ions come already in the excited state, although it's only some small portion of them. Then with the TIE sapphire laser, we tune to transition, and then we scan over these small shifts related to hyperfine structure and isotope shifts. And the signal is measured as fluorescence at this wavelength 541.915 nanometers. And in conclusion, while we perform this measurements of hyperfine structure of xenon with tunable TIE sapphire laser in the interval 820 to 841 nanometers, and identified wavelength regions around 820 nanometer, 841 nanometers, where transitions from 7 different xenon isotopes could be clearly identified. And I mentioned that for odd isotopes, since we have this isotope shift, the isotope shifts are determined from the position of centroids of these hyperfine structures that can be calculated. For frequency calibration, we use stabilized frequency comp reference to rubidium standard. And another technique for measuring this hyperfine structure is this fast ion beam laser spectroscopy that is presently also in development. And all this type of measurements can be used for measuring also isotope abundances, because different isotopes, they show these characteristics properties of hyperfine structure or isotope shifts. And that's all. Thank you very much for your attention.
Thank you. Now we're open for questions. We have a few more minutes. Okay, so then I will start. So in the spectra you got, do you have the information to actually get the dipole moment information for different transitions?
The displacements of hyperfine structure lines, they depend on this hyperfine structure constants, which in turn, if we have like xenon 129 or xenon 131, so there are two constants, one of them will depend on like interaction of electric field of electrons with quadrupole electric moment of the nucleus, and another on the interaction of magnetic field of electrons with dipole magnetic moment. So we get this information. Now, this in turn is related also to distribution of the charge of the nucleus. And so, of course, when we get this, we get additional information on these properties of the nuclei, but they can be also kind of correlated and used with other approaches for measuring these properties. So it's one of the methods that allows to gain this information. Sometimes it's a combination of certain constants, but certainly it helps to get additional information on these properties.
Sure, thank you. Any other questions from the audience? Okay, if not, we'll move on to the next talk. Alexander, he's going to give another talk, and the title is Local and Remote Measurements of Atmospheric trace gases.
Okay. Let me switch to another set of slides. Can you see my
Yeah,
screen?
but now we are seeing the presenter's screen.
Oh, presenter
Yeah,
screen.
you could share the other screen, then you will be.
Okay.
So you can stop sharing and then sharing the other screen.
Let me see. Okay, just a second. Well, if I move this to a different screen, let's see whether it will happen.
That may actually work. Yeah.
Let me try. No, it's probably not working. Yeah.
You have to start the presentation mode. Yeah, perfect.
It's okay. Yeah.
Yeah, perfect. Go ahead.
Okay, great. This is a talk about detection of presence of trace gases in atmospheric air and in some other gas mixtures. This work was done in group of Hans Schuster at Texas A&M University. Our graduate students, Carlos Rodriguez, James Bounds, and Eshta Alouawi, also involved in this research. And I should acknowledge contributions by our previous students and collaborators, Fiangshu, Aisha, Baiser, Jinbao Xia, Linbo Tian, and Sasa Zhang. The outline of my talk is that first I will consider cavity ring down spectroscopy for breath analysis, particularly detection of methane and acetone. Then another technique which is also allows sensitive detection is wavelength modulation spectroscopy. And here I will discuss local detection and also remote detection. And is in near infrared and also in the mid IR. And then a quite remarkable technique of dual frequency comb spectroscopy, which will be applied to detection of acetylene and methane. And finally, I will discuss some possible improvements of the detection limit by applying noise filtering and neural network also determination in multi-species gas mixtures. The minimal number of molecules in unit volume, this number density of molecules that can be detected is inversely proportional to the absorption cross-section of selected absorption line, the interaction length, and the signal to noise. So as a rule, we would like to have all these quantities large. For ring down spectroscopy, the principle is that we build up field in the cavity with a laser source, and then we abruptly turn off the laser source. So then we observe the decay of the field as it's leaking through one of the mirrors. And this ring down time is characterizing actually losses in the system. In this graph, we can see that red corresponds to empty cavity, and when we add some absorbing sample to the cavity, the decay happens faster. So the absorption coefficient can be related to this difference of the inverse ring down times of the cavity with the sample and empty cavity with this simple formula. And since absorption coefficient is related to number of molecules, absorption cross-section, and essentially concentration, so we can, from these measurements, get the concentration. If we have several absorbing species, then we have a system of equations where these coefficients A can be calculated from the spectral databases, and unknown are this is this vector of concentrations. And in the right-hand side, we have these differences of inverse ring down times. So from this system, if the number of measurements at different wavelengths exceeds the number of unknown species, we can get these concentrations using this formula, which gives least mean square fit to this system of equations. Now, this is a view of our experimental setup. We use a long chamber with cavity mirror separation of 172 centimeters, and reflective coefficients of these mirrors are very high, and they, as you can see, differ from 1 only in the 5th digit, which gives very long ring down time of 440 microseconds and very long interaction lengths of 132 kilometers. On this graph presented absorption properties of methane shown in red, and we see that there is a strong fundamental vibrational mode around 3.3 microns, and also another much weaker actually absorption band, which is like second overtone of this fundamental mode at around 1650 nanometers. So we use both of these absorption bands, and for near infrared like erbium doped fiber laser, frequency comb can be used, or some diode laser sources. And for 3.3 we used a difference frequency generation or interbed cascade laser, which correspond to these wavelengths. For measuring acetone in the near infrared, we can use this relatively strong absorption line at around 1672 nanometers. Here is the example of measurements of breath. On the left-hand side, it's a healthy individual, and on the right-hand side, it's an individual with suspected intestinal bacterial overgrowth. And methane, in the second case, has much higher concentration. And this is why actually methane can be one of biomarkers of different conditions related to intestine and colon cancer and so on. Here is example of measurements of acetone, and we have these measurements done on a subject with type 2 diabetes, and the spectrum was fitted with HITRAN database. So in the top, we see the result of the measurement, and below we see contributions of different components, like methane shown in red, water, which is shown in this blue, and CO2, it's this pink color. Acetone actually is this green line, and we see that it leads to displacement, to increase of the absorption, and rather like gradual this bump in the middle. Yeah. Now the concentration of acetone in this case is 1.8 parts per million, which is on the order of magnitude is quite typical number. Now, in the right-hand side, there is a series of measurements performed on a different individual, also with type 2 diabetes. And the results of measurements of acetone are compared to the concentration of glucose in blood. And we see that there is a pretty good correlation. The plot shows that this is almost linear dependence in this case. So this actually also suggests that acetone can be a good biomarker for diabetes. And also, we also know that for different individuals and depending on different actually conditions, these values can vary. Now I will describe our measurements with wavelength modulation spectroscopy. And this technique is also quite sensitive, although it's rather simple. And as the name suggests, it uses modulation of the wavelengths and also scanning of the middle wavelengths across the absorption line. As a result, we are getting signals at twice modulation frequency, 2F, and also at the modulation frequency, which can be detected with lock-in detection. And when we scan across the absorption line, typical shapes of the 2F signal is shown here, and 1F is shown below. And even better approach is actually to take the ratio of the signal at 2F to signal at 1F, because in this case, we suppress the influence of the laser power fluctuations. Here is one of the realizations of this setup. We use, in this case, local detection with multipass optical cavity, which has optical path of 290 meters. So the beam comes through a hole in one of the mirrors, bounces back and forth, and then comes out through the same hole. And this is a pattern of the reflection spots on these mirrors. Now the wavelength is tuned in this case by changing the current and also the temperature. And one of the results is shown below. So this is like Allen deviation analysis of the measured signal. And we see that minimal detection limit in this case corresponds to about 1.2 parts per billion at about 650 seconds, somewhere here. Also, this technique was realized in mid-IR at around 3.37 microns. It's more or less similar arrangement, except that the path length in this case was around about 580 meters. And of course, combination of the high absorption and long interaction paths give very low detection limit of about half a parts per billion. with 290 seconds integration time. Mid-IR also can be used for detection of acetone, and we made this measurement with this peak that is shown here. Our interband cascade laser overlapped this absorption line, but in this case we use very simple heliod cell with only 24 meters effective path length. So as a result, we got a detection limit of 1200ths of ppm with 60-second integration time. But of course, this is still sufficient for doing this analysis of breast samples, since we have seen that typical concentration about 2 ppm. Now, the wavelength modulation spectroscopy is also quite suitable for remote sensing, and we perform such measurements at the Release campus of Texas A&M on an old airport using one of the runways. So our sensor was positioned in one spot here, and then we use back reflectors that send the reflected light back. So the distance was 1.3 kilometers, and the total distance back and forth 2.6 kilometers. And for detection, we used this rather large telescopic system. So green light here is used just for adjustment, and we see this far spot somewhere which sends back the light. And in this case, we performed continuous monitoring for more than 24 hours. So this signal is shown here. It's around 2.5 ppm, which is somewhat exceeding average level of methane, which is probably justifiable for Texas. These large fluctuations are actually because of some passing cars were blocking from time to time our beam. And if we now in this measurement used also like reference channel with local reference cell filled with methane at 1% concentration, and the both signals, like the signal from the reference channel shown with black, and this red is our signal from our remote sensing, and these are actually spectra of the signals. And we can see that at low frequencies, so this peaks corresponds to the scanning rate, which was 2 Hertz in this case, and they are not present in the actual remote signal because of turbulence that suppresses these low frequency signals. Now, above about several hundred Hertz, the influence of turbulence already quite small, and we see this harmonics of the modulation frequency, which was at 20 kilohertz. And in this case, we got detection limit around 30 parts per billion with 60-second integration time. Now,
yeah, sorry to interrupt. We're
yes.
now a little bit over time. So if you,
okay, so I will wrap up. Yeah, I will wrap up. Of course, dual frequency comp spectroscopy is a very remarkable technique, and it uses these two frequency comps. which are detected on a detector and then with Fourier transform. So this technique is quite similar to usual Fourier transform. We are getting the spectra, which we are measured for essentially methacetylene and also for other gases, including methane. And one way to improve the signal is to use different types of filters. One of them can use, and it actually shows best performance based on neural sequence filter. And also neural networks can be quite efficient in determining concentration of different species in a mixture of gases, as was demonstrated with implementation of dual frequency comb spectroscopy and this type of processing of the results. And in this mixture, we measured 5 different components at ppm level with 1% accuracy. So this is the summary, and I appreciate and we appreciate the support from Welch Foundation, and thank you for your attention.
Okay, thank you so much. Maybe a very quick question, if there's any. Okay, so if not, I will suggest you to just message him directly in the chat. Okay, so so we're going to move on to the next talk. So the next talk will be given by Yanli Shi from Texas A&M, and his title is Transparency Resonances in Example of Germanium Vacancy Centers in Diamond. Please go ahead.
Hi, can you hear me?
Yes.
Okay,
you can speak a little bit louder.
Okay, can you see my screen now?
It's coming up. Yeah.
Okay. Hello everyone. I'm Yanli Shi. Today I'm going to talk about transparency resonances in ensembles of germanium vacancy centers in Diamond. This work is based on a recent experiment carried out in Texas A&M University by the group of Professor Alexei Akimov and the group of Professor Philip Hammer. So first, let's look at the advantages of GV diamonds. As compared to the rare earth-doped crystals, the GV centers has about 2 to 3 orders of magnitude higher dipole moment of the optical transition. And also it has a laminar system with a large energy level splitting in the ground state, which allows for orders of magnitude higher storage bandwidth compared to typically only megahertz bandwidth in the rare earth-doped crystals. As compared to NV centers, the GV centers has about 20 times stronger zero photon line, which results in more efficient single photon, single frequency interactions. And also, it has orders of magnitude less spectral diffusion, and inversion symmetry led to a smaller inhomogeneous bottoming. So the individual GV centers are well studied and has many promising applications, including quantum memories, quantum gates, quantum thermometries, and so on. And some of them could benefit from using ensembles of GV centers, because, for example, for realization of the ensemble-based quantum memories, the ensemble GV centers has orders of magnitude less spectral diffusion and smaller inhomogeneous bottoming compared to other ensemble NV centers, for example. And then, although the individual GV centers are well studied, less efforts have been done in the ensemble GV centers, where we have the inhomogeneity at level A here and level C. So our recent experiment shows that there is a homogeneity of about 26 gigahertz, which is much smaller than compared to the NV centers. And in the meantime, a relatively narrow and quite deep transparency net resonances were observed. But it still leaves us 2 questions. First one is, what is the origin of this resonance? And the second one is, since the optical inhomogeneity in level A typically is much larger than the low frequency inhomogeneity in level C. So we cannot directly see this low frequency inhomogeneity from our line shapes. Is there a way for us to extract this low frequency homogeneity from our experimental results? So here is our experimental setup. So we shine two 602 nanometer lasers to our sample, which is placed on the cold finger at 4K, and then we collect the fluorescence from this sample and filtered by a 16 nanometer long pass filter to filter out these reflections. The goal of the experiment was to find out the low frequency homogeneity WCB, and then since our probe and control laser are not perfectly aligned, so we need to take into consideration this overlap between these two laser spots, and also the proportionality constant K between rubber frequency and the square root power is not known, so we need to take into consideration these three parameters. So to solve the equation, to solve the problem, we need to solve the density matrix equations for the with the two inhomogeneities. And so we solve for the population as a function of this inhomogeneous detune delta AB and delta CB, and then we integrate it with the probability density functions, which should be Gaussian functions for inhomogeneous embodiments. But it is hard to find the analytical results, analytical expression for this Gaussian distribution. So we do the numerical calculation for the Gaussian distribution. But instead, when it counters to analytical formulas, we use a Lorentz distribution to find analytical formulas. When this decay from B to C level is 0, we find the population is just the same as the homogeneous Gordon case, but only to replace the coherence decays to these inhomogeneous broadenings. And our formula could work for arbitrary WBC, and in our case, the WBC equals to about 1/7 of WCB. And in our case, our formula gives similar results as in the WBC equals to 0 case. So here's our experiment results and the theoretical fittings. So the gray dots are the raw data, and the red dots is the moving average data for easier fitting. The thick black line is the our fittings using our formula. The dashed and the solid gray lines are our theoretical results under the same parameter, but with the 0 or full overlap. The green lines is simple fitting using two Gaussian to extract the window width, height, and position. So our fitting gives us the rubber frequency is about 3.6 gigahertz per square root of milliwatt times the square power. The overlap is around 40%, and the low frequency homogeneity is about 133 megahertz. On the left is our is a plot of visibility and width versus the rubber frequency. The circles is the experimental results. The solid lines is our theoretical fitting. Dash line is our experiment, our theoretical results used under the same parameter but with full overlap. So another question we need to solve is, well, what is the origin of such ensemble resonance? To find the answer, we take a look at each homogeneously broadened sub-ensembles. For example, if we look at the homogeneous sub-ensemble with 0 inhomogeneous detune, we find that their fluorescence line is just two sharp peaks with equal height, and it's symmetrically distributed around the resonance, which is clearly autotone splitting. And then when we increase this inhomogeneous detune to about 1 WAB, we found that one peak moves towards 0 and with sharply decreased height. When we add these up together, we find this consists of these transparency windows, and the other peak moves against 0. And when we take this into consideration, we find this is the wing part of the in our line shape. So we can conclude our works in two parts. First, we find the physical origin of the observed ensemble transparency windows is autotone splittings in each homogeneously broadened sub-ensembles. The summation of those resonance perfectly mimicked the IT resonance in the whole inhomogeneous ensembles. And such resonance could be used as similar to EIT and in the use of quantum storage and other promising applications. And also the second, our experiment estimates the inhomogeneous broadening at low frequency transition was about 133 megahertz. Thank you.
Great, thank you. Thank you for your talk, and thank you for making up the time. And so we have a few minutes for the question. Okay, I have a question. So in your modeling the inhomogeneous broadening of the two transitions, right? So
yes.
So the inhomogeneous broadening, so it is a Gaussian function for each inhomogeneous broadening, right? Or
yes, it's Gaussian in the numerical calculations, but we find it hard to find the analytical results. So we use the Lorentzian to do the analytical calculation for this integration.
But these two distribution functions, are they correlated or are they totally independent?
Actually, they should be a Gaussian for these two distributions because they are inhomogeneous, and then the Lorentzian can the distribution shape of the Lorentzian is similar to Gaussian. So if we use this Lorentzian distribution in the analytical calculation, we can have similar results as the numerical results from the Gaussian distribution, but with easier and more clear, like a formula for us to see what's happening.
Okay, but the two distributions are just random, right? They're not correlated. Which
no, they are just with same width.
Okay. Okay, thank you. Any more questions?
They have different widths, of course. They are not really correlated, yes. For the optical transition and for the low frequency transition.
Yes, these two are not correlated, but
they have different widths, of course.
Yeah.
The optical is very, very broad. And in fact, why we considered this question is because our experimentalist, Phil Hammer and Alexei Akimov, did the experiment and were asking us whether it is EIT or Otler towns. Because for Otler towns, they would expect that Rabi frequency should be larger than a homogeneous broadening to observe this splitting.
Mm-hmm.
But on the other hand, for EIT, you should have a large coherence time, but between these two sublevels, it is very short. And so we decided to have more careful look, and we found that actually Otler towns appears for different sub-ensembles in such a way that you still have a hole, because when you have Otler town splitting with the detuning, then the line which is closer to the two-photon resonance is weaker in amplitude, and that is how the hole is formed, as it is shown on this picture by Yan Li. So that's it, what it was about.
Okay, great. Thank you. Any more questions? Okay, if not, I would like to thank all of the speakers for this session, and we will conclude the session here. Thank you.
And thank you so much, Hibin. Just perfect. We finished on time. Everything great. Thank you so much to everybody who stayed with us, especially for those who stayed for the whole day. Thank you so much, and see you tomorrow. Tomorrow is the last day of our conference.
Sure.
Thank you. Bye.
See you, everyone.